Science is a way of knowing the natural world. In fact, the word science derives from a Latin root meaning to know. There are two main approaches, discovery science and hypothesis-driven science. Scientists, they often use a combination of these approaches to explore nature. Discovery science is the process of describing nature without any preconceived expectations. For example, Jane Goodall studied the social life of chimpanzees mainly by spending thousands of hours in the field watching them. You do this in your own life when, for example, you walk into a restaurant and note each of the different menu items available for selection. Like Jane Goodall, you are actually performing discovery science. You're observing the world around you and discovering what it contains. In discovery science, general principles are sometimes inferred from a set of specific observations. For example, Jane Goodall observed that some chimps strip twigs and then poke them into termite holes. From this, she concluded that humans are not the only animals to make tools. In your real-life example, after you note all of the food choices available in the restaurant, you may conclude that vegetarian options are rare. Many important scientific advances, such as the description and classification of organisms and the mapping of the human genome, have resulted from the discovery approach to scientific inquiry. In contrast, hypothesis-driven science involves constructing a specific, testable explanation for a phenomenon based on a set of observations. After testing this explanation, conclusions can be drawn depending on whether the results you obtain support or refute the explanation. Hypothesis-driven science typically follows a series of steps, these steps here called the scientific method. The scientific method is a loose set of guidelines for discovery intended to lead a scientist from an observation to an explanation. Have you ever had Peeps, sugary blob of sugar? My mother used to love these things. Okay, so observation is noting a fact or occurrence in the natural world. You notice that here in Northeast America, Peeps tend to get very abundant every year around April, and then they taper off in the summer, but then they reappear a little bit again in October. You incorporate some other observations from the natural world. Some animals reproduce on an annual basis. Animal populations can show an increase in number if you count them. So observations, they typically lead you to a question, which in this case you could ask, do Peeps have an annual breeding cycle that corresponds to temperature? A hypothesis is a testable explanation for your observations. It's important to note that a hypothesis is not a shot in the dark, but based on prior knowledge, on observations. Hypotheses are stated clearly. Peeps breed more with an increased temperature. This leads you to a prediction. Namely, if I go to a local peep breeding ground, I will see an increase in the peep population as it gets warmer in the spring. To test this prediction, you perform an experiment, an investigation of the hypothesis. I like to start with the dependent variable, although everyone tends to start with the independent. The dependent is pretty straightforward, because it's usually the data that you collect. When we write about experiments, students often forget to describe this one. Let's look at some of these hypotheses to understand how you might go about testing a statement that corn plants grow taller with fertilizer, right? You might grow corn in different concentrations of fertilizer, like 20%, 40%, and so on. And that makes sense. And then you might record the heights of the corn plants after letting them grow for a certain amount of time. What's the data that you collect? What are you measuring? Yes, the heights of the corn plants, right? And the data will allow you to say, yes, corn plants grow taller with fertilizer, or not, as the case may be. Let's skip to the third one that I have listed here. What is the data that you might be collecting? Price. Yes, this is what will indicate which jacket is cheaper. Before we do the remaining hypothesis, let's recognize something. Neither of these two hypotheses here tell you that height or price is the dependent variable. You had to think a little bit about how you would test these. Keep this in mind for the last one, the last hypothesis we're going to do. What might be the data that we could collect for this one? You might be saying, well, I would measure enzyme activity. Okay, yeah, I'll take that, but that's really vague. It's vague. Without really thinking about an experiment, we can't get more specific. For example, lactase digests lactose and produces glucose. You could measure the glucose, but that is so specific. This is why defining variables can sometimes be difficult. Sometimes they are vague. I mentioned this before, and this is what I mean by a controlled or managed experiment. In hypothesis-driven investigations, we define variables very specifically, right? And this variable is usually the most confusing to students. It's the independent variable. It's what's being tested. The independent variable is determined by the investigator and manipulated by the investigator to be different. So let's look at some hypotheses and try to determine the independent variable, which should also be the only thing that is being tested. Okay, corn plants grow taller with fertilizer. I'm not debating this with you. It's just an example, right? What are we testing? You might say we're testing the corn plants. Okay, but wrong. Let's think of what we might do to test this statement. We might grow corn in 2 fields, use fertilizer in one and not the other. You're varying, you're manipulating the fertilizer, right? You're determining the concentrations of the fertilizer. This is what you are testing. You are testing the effect of the fertilizer. All right, let's think about the next statement, right? Because practice is the way to understand. We could use lactase, which is an enzyme, and it digests lactose to test this, right? You might put lactase with some lactose in different temperatures, and then you measure something that indicates how fast they're working. What are you intentionally manipulating to be different? It's the temperature. That's what you're testing. All right, let's do the last one. How would you go about testing if store X has the cheapest price for the jacket that you want? You might go to store Y and store Z and store A and write down the price that each store offers. And then you're choosing to go to these different stores. The stores are the independent variable. Let's recognize something here for the independent variable. You might want to define it as the thing that is being tested. And this statement usually leads you to the dependent variable, the height of the plant, the price of the jacket. This is why you have to ask, what would I be intentionally manipulating to set up this experiment? Controlled variables they're usually pretty easy for students to recognize. These are usually, or there are many ways of manipulating them, and they're all constant, like using the same corn plants, the same enzyme, the same jacket. The problem with controlled variables is that they get confused with what is called the control. All right, so the control is different than the controlled variables. Please, 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 please know that right here at the outset. The control is a basis of comparison. This is really hard to define for some experiments. Let's start with the easy ones. We might amend this first experiment with corn plants so that we're growing plants with different concentrations of fertilizer. We'd need some plants grown with no fertilizer as a basis of comparison. So plants not grown with fertilizer here are the control. Okay. Yeah, this one is tough. What do we designate as the control for temperature? 0? Room temperature? Body temperature? You know what? You get to decide a little bit, but you better be pretty clear about why you want to use it as the basis of comparison. How about this? When you set up this experiment, you combine the enzyme with some other goo, and it all starts out at the same temperature. Maybe that's the basis of comparison. I know, a little bit tougher. Are you thinking about a placebo group or a group that you didn't give to drug X to? Yes. Yes, that's the control group. So these can be confusing. When you encounter these concepts in later labs, I urge you to come back to this presentation and review it. Comparing your experiment to these examples can be the way for you to answer the questions. As we went through these variables, the definitions, they don't really help, right? It's the examples that help. Those last 3 slides about variables, they're sometimes better understood if you think of a graph. If you're having trouble thinking about an experiment itself, you might be able to think or look at a hypothesis and think about what kind of graph you might make as an outcome. That's good, because the dependent variable is usually on the y or vertical axis. And that's easy enough, right? Think about the corn plant height, the jacket price. The independent variable is also usually along the x or horizontal axis. Let's go back and let's think about the peeps. We wanted to see if they were produced more in the spring. So we determined to measure at different times during the year. We manipulated that. We determined that, so that's our independent variable, and yes, it is on the x-axis here. We counted peeps each time we did our measurement, which is on the y-axis too. It's important to let your data determine the right type of graph to use. What would be best to show your conclusion? Our graph here shows that peep population increases to a peak in April and then decreases until a small bump in October. And we can compare this to our expectation with our hypothesis. Before we go on, I want to point something out. Many students who take this class are business majors and education majors, and in business, you're often creating graphs as displays of data, and there are different rules in business than in science. So if you are a business major, you might want to default to a bar graph, and you might want to put the independent variable along the vertical. I appreciate that because it means you've been well trained by your business instructors to do that. But we here in this class, we apply a different display. So check out the graph on the left. I mean, really look at it. The x-axis, it doesn't make any sense, right? You don't have the ability to rearrange those numbers like that. You can't do that. They have a predetermined sequence. Think of something like temperature or age. You can't reorder the numbers. When this is true, a line graph is the way to go. Could I have used a bar graph here for the increasing height of the child? I could, but it's better to use a bar graph here. Let's look at the bar graph on your right. The meat along this is a vegetarian's nightmare, right? The meat along the x-axis, they don't have any predetermined order. You want to do them alphabetical? Go ahead. Some people might not. They want to be like, well, I like beef, and I'll do that first. I could have gone the alphabetical route with them. But since they have no predetermined order, I decided the order of them, and I liked this order because it showed a nice increase in the bars. Maybe you like a decrease. That's fine. That's fine. But with the bar graph here, we have choices, unlike the line graph. So this is why we use a bar graph, because the independent variable along the x-axis, it doesn't have a predetermined sequence. I can rearrange it. I want to ask you to be aware of this when you have the choice and make a good choice. Don't just slap stuff up on a graph. You got to take a step back and ask, can I make this prettier? Seriously, like, presentation counts here. Oh, all right, getting back to our peeps again. Once we've generated the data and we make a pretty display, we have to interpret it. We can't just say, here's the data, you figure it out. We have to like narrate the data. You have to think about your textbooks when they present graphs. The graph is referred to in the text and it's usually explained. We have to do the same thing. Before we get started, there's something that is oh so useful to do. You got to remind yourself what it was that you set out to do. Because sometimes at this point, we forget. Our hypothesis was that peeps reproduce more in the spring. Our data partially supports our hypothesis, and that's okay. As temperature rises in the transition from winter to spring, peep populations increase, yes. However, they decrease in the summer as temperature continues to rise, and then there's this small bump in October, which could be due to a brief temperature burst commonly known as an Indian summer, which I think is now a politically incorrect term. Retraction. As a student, rejecting your hypothesis is just as important as accepting it. When accepting your hypothesis, it's important to suggest the next experiment to keep the overall investigation going. Is there an optimal temperature for peep reproduction? Is that why we see 2 increases in the population in the year? When your results don't support your hypothesis, which is often the case in science, you can revise your hypothesis or construct a new one and then repeat the method. You may thus end up running through the scientific method several times in order to explain the observation that began your inquiry. As you may have surmised, you actually use both discovery and hypothesis-driven science every day, though you may not think of it as such. If you pay attention, you'll find yourself following the scientific method many times in your daily life. Therefore, you already have a lot of experience with these types of inquiry, and you're kind of on your way already to understanding that scientific process and those steps. You should really check out peepresearch.org. Although a ridiculous application of the scientific method, the mock experiments on the website carefully use the steps of the scientific method. So it's kind of just a reminder that the scientific method is only a way of knowing, and in our case, a way of knowing peeps.